Welcome to this first invited session on statistical methods for neuroimaging studies of Alzheimer's disease. Forgive me if I'm a little bit slow as session chair today, but it is like 6 a.m. over here on the West Coast. We do have a really amazing session that was organized by Tudorascu. I will do my best to keep things on track as well as possible. We've got three great speakers, Ciprian, Donna, and Joanne Beer. And the talks will be 20 minutes each, and then at the end we'll have the floor session. So if there's time for questions, we'll have a question. Otherwise, we'll just move on straight to the next talk. Let me see. So if you have any questions, please put them in the chat, or you can raise your hand during the floor session, and I'll try and moderate the questions somehow. So without any further ado, I'd like to introduce Ciprian Crainiceanu for the first talk on longitudinal image analysis and inference. And Ciprian is a professor at Johns Hopkins University.
Can you see my screen?
Yes.
Is it? Okay. Well, I just wanted to say that I know that this was room A, and just wanted to say that there is a room B. Please have a look at the room B, so that if that's something that you are interested in, you are not just staying here because you were here. The other parallel session is a fantastic session as well. So if you want to take time, come in and out, that's also fine. Just wanted to make sure that you know about this second session as well. So today I decided to talk about a couple of very simple ideas that I've had. So over the years, I worked a lot on complicated things, and this is still complicated, but I was thinking about something very easy, something that at the end of the talk, people can take and say, you know what, I can implement that. So here is the problem. So the problem is that we are trying to measure longitudinally images. In this case, the images that we have are a particular part of the brain. In this case, it's corpus callosum. And what you see here, these are some manually placed landmarks. And between these landmarks, there are equally spaced segments. So this is the posterior part of the brain, and this is the anterior part of the brain. So you go from the back of the brain towards the front of the brain. This is the corpus callosum, is the fiber, the collection of fibers that are in the middle of the brain and connect the two hemispheres. So one of the things that we wanted to do is to measure along the center of this corpus callosum. We want to measure fractional anisotropy, which is a measure of water diffusion in the brain. But we can do this repeatedly. In imaging, we can do this repeatedly. So over the course of about 4 years, we see this image multiple times. So the same person comes multiple times, and every time we measure the fractional anisotropy, which is a measure between 0 and 1. 0 meaning basically water molecules diffuse at random, and 1 means they diffuse along a particular track, right? So that gives rise to our data. So the data is, in this case, 0 to 120. This is cut a little bit shorter here. And every line here, is 1 visit, right? So you may start to see some things that you don't like. For example, this is a person, this is another person, so this is a person with 5 visits. For example, just looking at the data, you see that there are these 3 visits, and then there was visit 2 that was much, much lower in terms of fractional anisotropy. This is not biologically believable. It is not biologically believable that somebody dropped so much in their fractional isotropy, then bounced back again. So when I looked at the data, I was a little bit worried about these type of things. But fundamentally, that's what we have. We have hundreds of individuals over 5 years. All of them have multiple sclerosis. Actually, we also have people without multiple sclerosis, but here I'm going to focus only on the subset of individuals who do have multiple sclerosis. So that's what we have. We have data that is longitudinal in nature. And of course, you can make this as complicated as you want. You don't have to take the corpus callosum. You don't have to take a function. You don't have to simplify it. You can go to the entire brain. So everything I'm going to talk about, it's about longitudinal image analysis. It doesn't have to be just about the corpus callosum. Now the question is, I'm in my office and I look at this data, it's complicated, and the question is, how do I analyze it? And I propose the following very simple thing. I propose for just a little bit to ignore the location, these S's, right? Ignore the location and just fit a pointwise linear mixed effects model, right? So what you see here is a fancy way of saying I'm going to fit a pointwise linear mixed effects model. And the nice thing is that these YIJs, they don't have to be continuous. They don't have to be, they could be Poisson, they could be Bernoulli, they could be anything. So for example, you could have things like presence or absence of a lesion and so on and so forth. So this is, and we are all very familiar with this type of models. We know how to do them for a fixed S. What I'm saying is, well, do it for one fixed S and do it then for all fixed S's and see what happens. And then the second step is actually not necessary, but nice, as you'll see in just a moment, where you calculate the fixed effects, the random effects, and then you smooth them. Okay, so we should all know by now that all smoothers that I know of are of the type S, which is a matrix, times the vector. So a lot of the discussion in smoothing and non-parametrics and stuff like that is what S is better than the other S's. In this talk, I don't care about that discussion at all. It's just one S that smooths the data. So whichever you like. And now this is the kicker. How do we do, of course we can do pointwise estimation, but how can we do confidence intervals? And this is where the idea is, again, very, very simple. Bootstrap study participants and calculate the pointwise confidence intervals for this. And then use what is called a leave one in bootstrap. I mean, what I call the leave one in bootstrap, where you sample the data and you always leave in the person for which you are interested in doing inference about their random effect. So that's it. That's the whole procedure. It's simple. It can be easily parallelized, and it scales up very easily to. We applied it up to 10,000 observations per person. It can easily be parallelized to millions. So let's see what happens in the data. This was the original data, and then we applied a pointwise. So at every point, we applied a linear mixed effects model. And lo and behold, very nicely, all of a sudden this says, you know what? A lot of this is measurement error. So I'm just going to shrink all of them towards these estimators. And this is for the second individual. So it says, no, this is actually measurement error. I'm just going to shrink it. It's not there. Very nicely done. Exactly what mixed effects models are supposed to do. And then, of course, when we submitted the paper, there was a lot of discussion. Well, how about the smoothing? You see that the smoothing, at least in this case, doesn't do that much. The smoothing done by the mixed effects models does most of the heavy lifting. And then you can additionally smooth over the domain if you want, but it doesn't do that much in this situation. So it varies by application, of course, if you have more noise or less noise. But what you see here is just the S matrix multiplied with this. Anyway, so I hope I convinced that this is a reasonable idea. Then we looked and we did inference. So what you see here, these are the confidence intervals in gray and the joint confidence interval in light gray. And you may ask, well, why don't you just use, Bonferroni correction or FDR or all of these things? Well, let me remind you that you can sample these things as closely as possible, so the Bonferroni corrections will explode to infinity. Okay? So because we are in continuous space, you may get lucky with a particular type of Bonferroni correction you make, but if you go from 1 millimeter to 1 micrometer, the inference is not consistent with FDR on Bonferroni correction. Just want to put that out there. This is the big difference between functional or imaging data and other types of data, which is that you can keep on sampling at lower and lower dimensions. But here is the most interesting thing that I wanted to show you. So there are no gender effects in our case, no sex effects in our case. There is some age effect, but not much. But this is the big effect that we found. This is scan date. So this is years from the scan date. So there is a 0.02 effect on the fractional isotropy per year. So over 5 years, you get about 0.1. So 0.1, let's have a look at this. 0.1 on this scale is about this much. So in 10 years, you can get about this much of an effect from the scanner. So where is this coming from? This shouldn't be there, right? It shouldn't be there. What happens, we are looking at technology getting better. That's what's happening. Technology getting better makes FA larger across the entire population. So if you want batch effects, substantial batch effects, you see right here. So for me, this is just a reminder that we should not necessarily take imaging data without carefully thinking about batch effects. So this is showing how to get the joint confidence intervals. Essentially, the idea is to treat the beta parameters as a multivariate normal, then sample from that multivariate normal and take the maximum over all differences over the t statistics, and that gives you essentially the distribution. So you can get those joint confidence intervals. This is consistent with subsampling and things like that. We compared our approach with the fraction additive mixed effects model, Fabian, Scheipel et al., which I think is the state of the art in this area. And indeed, our very simple procedure matches what they do in terms of number of subjects. So this is integrated square error in terms of, so this is also when the number of visits increases, and this is the dimensional functional domain. We had to stop at 400 for FAMM because FAMM, as you see in a moment, this is the time in hours it takes to fit these models. So here, our method, the pointwise plus bootstrap, is doesn't change that much. And at 400, the FAMM starts to explode. It takes more than 10, 12 hours, and we were not able to do 500 and 1,000 because that's infinity in my time. So it works. It works. You can do it, and it's a very powerful technique. So also compare confidence intervals. We do quite well. The last thing I wanted to talk about is a seemingly unrelated problem, which is the abstract. So we all are familiar with the bootstrap, but for some reason, I and maybe others thought that, well, bootstrap is set in stone. You can only bootstrap as many subjects as you have data. And I thought, why? Why in the world can we not sample more or fewer observations when we do the non-parametric bootstrap? And in fact, it's a really good idea. So what are the advantages? Well, you can analyze the behavior of estimators in small, moderate, and large sample sizes. Does not require explicit modeling of complex associations or correlations. You can calculate sample size in complex data sets, which you cannot do. We know that sample sizes beyond t-test is almost impossible without. So looking at this data, I could ask questions of the type, when will age become significant, or with probability 90%, if I increase my sample size? So I can resample my data with more sample size and then look at how these confidence intervals decrease. Or I could ask, is this a line, and have a test for the line and increase the number of subjects, and so on and so forth. And this can be applied for any type of test statistic that we want, especially in our complex. So here is an example. We have in the sleep heart health study, we were looking at the association between moderate to severe sleep apnea. And we were looking at the effect of hypertension on sleep apnea. And the p-value was 0.109. And the question is, well, what sample size can would we need for hypertension to be significant in 90% of the time? And then you can do the abstract, so you can sample more. So this is twice the sample size, 3 times the sample size. And you see that the effect gets significant at 80% at about 3 times the sample size. Anyway, I think that this is an interesting combination of things, and thank you for your attention.
So I'd like to welcome the next speaker. Donna Tudorascu is a professor at University of Pittsburgh, and her talk is Methods for MRI and PET Neuroimaging Data Harmonization across different scanners in Alzheimer's disease. Thanks, Donna.
Thank you, John, and thank you, Ian, for the invitation. Can you guys see my screen okay? Okay.
Yes, perfect.
Thank you. So I would like to start with a little bit of overview of what I'm going to talk about today. So I'm going to touch upon some challenges that we encounter while analyzing neuroimaging data from multiple scanners or multi-centers. I will talk about some methods that we have applied. These are existing methods, not new methods that we have developed. So we looked at Ravel and Combat and a combination of the two. And I'm going to go over some of our results. So the motivation for this specific work started a couple of years ago when we were trying to integrate some of the data that we have acquired on our Alzheimer disease imaging studies here. And some of the subjects were scanned on 1.5T and some of the subjects were scanned on a 3T. So we were looking to characterize the disease progression longitudinally, but we were also looking at cross-sectional data. And as subjects came in, we switched from one scanner to the other. So we had data at baseline collected on different scanners. Then more recently, we started working on these big consortium studies, which are multi-center studies that have acquired data on multiple scanners. They acquired different longitudinal neuroimaging modalities longitudinally, and we also have very many sites that are participating. And one of them is the Alzheimer Biomarker Consortium in the Down syndrome. And then the other one that we just started is a study on vascular cognitive impairment and dementia with UC Davis, specifically on why matter lesion etiology of dementia in diverse population. This is also a longitudinal study. It also has PET imaging, MRI imaging, multiple center, multiple sites, different scanners. So all sources of variability. The goal of these studies specifically is to characterize disease progression using these neuroimaging markers that are derived from PET amyloid, tau, MRIs, and the end goal is actually to contribute to the development of treatments in Alzheimer's disease and vascular dementia or other brain disorders. So our role as statisticians, and specifically my role and our group role for these studies that I'm referring to, is to provide some type of accurate quantification or integration of these imaging studies and analysis, statistical analysis for all these collected biomarkers for imaging. Oops. So there are different scenarios for combining imaging data or data from different scanners. So you can have data from different scanners with different magnet strengths, such as 1.5T, 3T, and most recently we started with 7T data. You can also have data that are from different scanners, from the scanners with the same strength, but you have different manufacturers. So you have GE, Philips, Prisma, and other manufacturers, and those are very different. You can also have data from scanners from the same manufacturers as we have from some of our PET studies from the ABCDS, but at different institutions. So because you're studying specific population, and these are not clinical trials, but observational studies, the heterogeneity is different and the sample is different at different sites. So even if you have different scanners, you may end up with differences because the population that you're studying is also very different. So we started with a very simple experiment, and we were thinking, what are the actual main differences between a 1.5T and 3T scanner? Is that the fact that the 3T scan has approximately twice the signal-to-noise ratio of the 1.5T, and when we try to segment these images, the contrast is going to be very different, because what our outcome is actually at the end is the gray matter, white matter, or CSF. And these tissue types are required, the brain images are required at the very, improved contrast of the 3T compared to 1.5T. So combining these data could be very challenging. So it can lead to major differences, such as the segmentation, the differential PET and MRI co-registration. So for those of you that are working with PET imaging, you know that PET has a very low resolution. So in order to find those imaging modalities and ROIs and combine them, you will actually need an MRI for the PET MRI co-registration. And these imaging intensities can vary substantially between different magnets. So what we have to do is to minimize or eliminate as many sources of variability as possible. So statistically, we can do that by design, and we're good at doing that with the clinical trials, but we're not very good at doing that with these observational studies. And then what we're trying to do is to eliminate basically these scanner differences so that the measurements are as accurate as possible. So we were very lucky here at Pittsburgh to have a sample of 16 elderly subjects that were scanned on 1.5T GE scanners, and then they were scanned on a 3T scanner, and they were scanned about 3 months apart. So whatever happened in their brain, that was not happening because of the disease. So within 3 months period, the anatomy of the brain shouldn't change very much. So if there were differences between these measurements or in the actual gray matter, white matter, it is actually due to the scanner differences. So this was a sample of, as I said, of elderly. The median age was 77, and with a range between 70 and 79, about 25% were males. So we just started with this investigation to see what are the actual differences. So we've done some basic segmentation, and we performed that using SPM and FreeSurfer, and we just looked at the gray matter volume. So we look at between scanners within each software and within scanner between software, just to see how the data look like and what can we do to improve it. So now, when we just looked at the 1.5T and 3T, just density, plotting the intensity values from the 1.5T, which is actually in blue here, and the 3T in red, they were very different. And these are the same subjects. And when you look at this, you would expect them to be the same. Now, I'm not going to talk about actual software differences, but just looking at the FreeSurfer segmentation of the gray matter volume, you can see that for the 1.5T was 622 millimeters, and then for the 3T was 526. Now, for the SPM was kind of reversed, but the idea here is that for the 3T and the 1.5T for the same brains, we have seen very, about 96 cubic millimeters differences between the same subjects between the two scanners. So that was kind of huge. And then when we looked at the, just the ICCs, they were very, very low. So we wanted to apply some of the existing methods for this. So the existing methods in the literature that were previously developed for this kind of combination and accounting for these kind of differences were Ravel and Combat. So we investigated first Ravel. So for those of you who are not familiar with the method, there is a paper that was published in 2016. This is work that was done by Taki and the student at the time, JP. And this Ravel is removal of artificial voxel effect by linear regression. So it's basically an analytic tool that is designed for the removal of residual technical variability. It has its roots in SVA and genetics, because the genetic field is, I would say, way ahead of us in terms of this, because they always control and they have these batch differences when they process the data. So Ravel has two steps. So the first one is you conduct a factor analysis on the control voxels. So the control voxels are the ones that are considered to not be related to the disease. And in this case, you're looking at the ventricles for Alzheimer disease to estimate the principal directions of technical variability. And then the second step is to conduct a linear regression at the brain level that includes both the covariates of biological interest and the direction of this unwanted variation. So the actual end goal is to obtain an analysis about these relevant predictors while accounting for artificial variability. And as I said, there is a paper on specifically on Ravel. And then the combat, that is data harmonization technique, while Ravel, you could see it more as an intensity normalization technique. So combat is a method in which the data within each site and scanner is first modeled as a linear combination of the biological variables of interest and the scanner effects as additive and multiplicative in the model, and then is adjusted to be harmonized across sites and scanners. So this is the actual model of the Ravel, and our focus was to harmonize these measures. So we are looking at the cortical thickness and the volumes, so that were extracted from these raw images. So you extract this and then you model them. So the combat is actually a method that is done for regions of interest of the brain, while Ravel is applied at the voxel level. So if we, so a little bit of a pipeline that we have actually used for this combination. So we have the raw, what we call the raw images are the images that were collected from the scanner. So we perform free surfer on those images, and we also got the summary measures, and then we have performed combat on these measures. And then we took the raw images, we performed Ravel, the intensity normalization, we extracted the Ravel normalized images, we have done then a free surfer segmentation, and then we call these measures the Ravel normalized. And then the next step was to take the raw images and to perform Ravel and then perform free surfer. And then we use combat, and then this is the combination with Ravel and combat. So to sort of like figure out if Ravel was working, because we actually don't know what the truth is here. So the best thing that we had, the best tool that we had, was to have a neuroradiologist and look at some of the regions that were segmented after we applied this, the intensity normalization, to see how accurate these regions were segmented. So we've done some visual ratings for hippocampus. So we only had one neuroradiologist that looked at this. The neuroradiologist didn't know what method we actually used. So if it was just the raw images that were segmented with free surfer, or if it was the raw images that were segmented with Ravel. So, and they had a 4 rating scale for segmentation. 1 was poor, 2 had some errors, 3 was good, and 4 was excellent. So if we look at some of these plots of the intensity values for the raw images, so you can see, so the 3T scanner is cyan and the 1.5T scanner is the orange one. So if we look at some of these are from the raw, extracted from the raw data, without performing any type of intensity normalization. As you can see, they are very different. The orange right here and the blue are very, very high compared to those. Then the white stripe normalization, which is a step of the Ravel, it is also an intensity normalization technique. It makes the data looks better. And then these white stripe intensity normalized images are fed into the Ravel, and then these are the Ravel normalized images. So in terms of intensity and how the distribution looks like, they seem to be pretty good by comparing with the raw images. So this is a table with just the descriptive statistics. The main idea here is to see that there are differences between the raw, the 1.5T and the 3T data, even though the differences are small here, but also the values that we're looking at are very small. So we've done this for the cortical thickness, we've done this for the volume, and as you can see, some of these regions, for example, if you look at the entorhinal, so the value is 3 for the cortical thickness for the 1.5T and 3.24 for the raw data for the 3T. They are a little bit closer when you look at the combat harmonization, 3.08, 3.16, and then Ravel combat, that difference is actually a little higher and the variability is higher. So if we actually look at the mean and the standard deviation of the cross-scanner differences or bias and the RMSEs, and what I want to show here is basically, so the values that are shown in bold are the values where the RMSE is actually increasing compared with the raw. So the idea to look at this was to see if Ravel is performing better than the raw, if combat is performing better than the raw, and the combination of the two is performing better than the raw. So in all of these comparisons, the RMSEs for Ravel has different areas of the brain for the cortical thickness that are increasing, 4 of them, and then for the volume that are increasing. In terms of combat, the bias is smaller, closer to 0. The RMSD is smaller, and there are only 2 instances for which RMSD is actually higher compared to the raw. And for the Ravel combat, the RMSE is higher for multiple instances, 4 here and another 5 times for the volumes. So judging by this, so combat is actually the one that performed the best with the smaller mean differences, with the smaller RMSD for all these cortical thickness and volume measurements. So if we look at the neuroradiological ratings, we can see that there were differences between raw and Ravel with respect to the lower ratings for the left hemisphere of the hippocampus. However, the ratings weren't that different with the right hemisphere. If you look at the ones that perform in the 3 that were good, so we have 29 for the raw that were doing better than compared to the 26 with the Ravel. And here for the right hemisphere, we have 29 of the Ravel and 28 of the raw. And then these are just some images that are showing us that, for example, for the hippocampus segmentation, for Ravel normalized images in red is more accurate than that of the raw images that are in green. So this is when we use Ravel, this is more accurate as the green area, it's including some extraneous segmented voxels over the CSF or some of the adjacent areas. So in this image, though, the raw performed better as compared to the Ravel. So the Ravel is actually in red and is less accurate because it fails to include some of these areas of the hippocampus. So therefore, we also looked at some of this for the PET imaging data. The only thing that I wanted to say that with respect to PET SUVRs, because those are different types of measurements and are obtained differently, we didn't find much differences between the Ravel and no Ravel, and we looked at the amyloid negative and amyloid positive subjects here. So in terms of the effect sizes weren't that different between when we used the Ravel and when we didn't use it. As compared to the cortical thickness, they were much more different. So PET seems to be a little bit more stable than the MRI. And then what I would like to say is that we have to be very careful when we consider scanner effects when combining studies from different scanners and different sites. We've seen that combining an intensity normalization with the ROI harmonization provided better results compared to intensity normalization only or no adjustment, that actually combat seems to be superior in terms of scanner harmonization. So our previous work shown that with respect to PET, the SUVR, the differences were smaller, but you can make a higher impact when you classify subjects. And MRI measures are much more different with respect to scanner differences compared to the PET. So this is the work that the graduate student that is working with me had performed, so she should take all the credit for this. These are some of the collaborators which I would like to thank, and Ciprian and John at Hopkins and the University of Wisconsin, at UC Davis and Baltimore, and some of the references. And thank you.
The final presentation for today is from Joanne Beer, who's a postdoctoral researcher at UPenn, and look, if you want to get your presentation up, Joanne.
Sure.
And I am going to.
Oh.
Like I said, it's early in the morning here, so please forgive me for not having the talk titles up in front of me.
It's okay. It says host disabled participant screen sharing when I click that.
Oh.
There's a way to fix
I'm pretty sure I didn't, but that's, I suspect it's the organizer somehow, but I'll see if I can figure out. So security, I think is where I have to look. It says participants are allowed to share screen.
Is it?
Yeah.
Yeah. Brandon, can you please make John the co-host, please?
Well, I'll announce the talk title at least while that's happening. Joanne's talk is on the extension of combat for harmonization of multi-scanner neuroimaging data in an Alzheimer's disease neuroimaging initiative dataset. So Thanks a lot, Joanne.
Thank you. And thanks to Dana for the invitation and all the other organizers. So Dana kind of covered a lot of, she kind of touched on the topic of harmonization. So so I might skip some things, but yeah, as most people here might be familiar with, there are batch effects, so-called batch effects that occur in multi-site or multi-scanner neuroimaging data. And just for the sake of definition, we can consider batch effects to be unwanted technical as opposed to biological variability related to the site or scanner on which the data was acquired. And data harmonization is like removal of this unwanted variability. Yes, and then we also have studies that look at the same participants over time. So scanners might be changing over time within a given site as well. And here's just an example of the many multi-site studies that are now going on. And these are really great because they can yield greater statistical power. But we have the challenge of dealing with these site and scanner effects. And here's just an example from the Baltimore Longitudinal Study of Aging, where we have the same participant scanned on a 1.5 Tesla GE and then a 3 Tesla Philips scanner. And you can see the differences in the, the intensities in the image. And so these differences sort of translate even to when we extract measures, like let's say cortical thickness measures. Here's an example of in the ADNI data set, this is the Alzheimer's Disease Neuroimaging Initiative, an example of one region of interest that had the largest shifts in the mean for the cortical thickness values. And you can see that the 3 Tesla scanners in the red really produce inflated or higher values for cortical thicknesses than the 1.5 Tesla scanners. And we found in this ADNI data that a lot of these scanner effects were like in the medial temporal. They were strongest in sort of the medial temporal region or occipital region. And there are also differences in the variance. So here's a picture of the multiplicative scanner effects. So we can see the largest effects are coming from a specific vendor in the pink. There was just more error variance after we adjust for, age, sex, diagnosis, and time. There's been a lot of studies into what the potential causes of these site and scanner effects are, and can be related to, anything from the subject characteristics to acquisition protocols, scanner hardware, magnetic field and homogeneities, and so on. So when we adjust for scanner effects, though, we're often just kind of agnostic to a lot of these differences. Just thinking about how to address site and scanner effects across the whole data acquisition and processing pipeline, it's good to address them at every stage of the study and the analysis. So this is just sort of a taxonomy that I came up with my postdoc mentors, Kristen and Taki, helped sort of think about this. So, first of all, you want to standardize acquisition as much as possible, including the study sample across the different sites and scanners and protocols, possibly the scanner manufacturer and models. There's also ways to incorporate traveling subjects. Intensity normalization is something done at the image level rather than, let's say, at the feature level, like extracting feature, but at the image level. But it usually does not explicitly account for the batch in the normalization model. And then what we are calling statistical harmonization does explicitly account for batch, and it can be done both at the image level or after feature extraction. So let's say we extract the cortical thicknesses from a set of regions of interest. Combat would be an example of this. And then we can also do robust downstream analyses, meta-analysis or mega-analysis, for example. So just a point that study design is important. So even when using combat and so on, we want to be sure, like, for example, diagnoses and other, age and sex are balanced across the different batches, ideally. So I'm just going to focus now on some methods, some combat methods and extensions of combat. Combat stands for combating batch effects when combining batches. I'm not sure really who developed that acronym actually, but because this original paper does not call it combat, from what I can remember it. But it was originally developed for genomics data. We have this 2007 biostatistics paper, and it harmonizes the means and variances of the residuals across batches. So it's a form of location scale harmonization. And you can include covariates to specify variants you want to preserve, such as age, sex, and diagnosis. And it uses an empirical Bayes estimation method that pools information across features. So, combat's designed for when you have multiple measures on a given subject, let's say, so regions of interest or voxels or whatever. And it is, more robust to outliers in small batch samples. So we're assuming that we assume that within a given batch, the additive and multiplicative batch effects across the features, denoted by the V here, are deriving from a common distribution. So like the additive batch effects, we can impose like a normal prior distribution, and then for multiplicative batch effects, we can assume an inverse gamma distribution, and then we get shrinkage towards the mean, so the overall batch mean for the means and variances. And an example of why combat might be preferred to just simply a least squares approach is that we can preserve the sort of the relationships better when we have these small sample sizes and we have like an outlier here. So the combat adjusted data preserves the pattern, the relationship better than the least squares. And here's just sort of the model. So we assume the side effects exist in the mean and variance of the residuals and then we have multivariate observations features. And the additive batch effects is present in this gamma, and then the multiplicative effects are this delta here. And then we have our covariates that we want to preserve in the X beta. And yeah, as I mentioned before, we assume these priors, and then we can do our estimation, and then we can remove the batch effects from the data while then adding back the biological variability. So it's been possible to extend this combat methodology to like different types of models. So yeah, like J.P. Fortin and other people in Pensive sort of worked on originally adapting combat to neuroimaging, and Mei-Chin Yu did an example with functional MRI data. And this is a paper that came out last year. We did on longitudinal combat. Ray Pomponio and Christos Davatsiko's group at Penn has developed the combat for generalized additive models. And I'm working on generalized additive mixed models using the R package CAM4. And then Andrew Chen and Hao Chang Xiao have developed CoveBat for harmonizing feature covariance. And this one, I didn't fill in the detail here, but this is for functional neuroimaging data. And Andrew Chen has a poster on that in this conference. So I'll mention that later. So this is just some details about the ADNI data set that we used for the longitudinal combat, around 600 participants. And this is kind of like what the data looks like in one region across different sites. And in different sites, they have different scanners. And the scanners are possibly changing over time within a given participant. And here's an example of what the data look like before and after longitudinal combat harmonization in the left fusiform. An area that shows a lot of atrophy, typically in Alzheimer's disease. So you can see a lot of the variability is removed after harmonization. And here's a picture in the form of box plots of the adjusted residuals. So we did statistical testing to show that there were no longer mean or variance differences across the scanners after combat. And here's, the model for the longitudinal combat. So it's basically the same as combat, but we add in subject level random effect. You can also add random slopes or other random effects. And from our simulation studies, we saw that the longitudinal combat adjusted data, which is in the blue, I'm kind of comparing that to the orange here, which is just unharmonized data with, let's say, a scanner covariate, which would be the dark orange. And we find that the estimates, the means are very similar as far as like the bias and the standard errors might be a little smaller after longitudinal combat. But I guess a good feature is that it's preserving the type 1 error, the nominal rate, better than unharmonized data was in the ADNI or in this simulation, I guess, which, and this is sort of under the alternative hypothesis with strong, moderate, and weak effect sizes. And here we see, the p-values, higher would be smaller p-values, so there's minus log 10 p-values. And we see that the longitudinal combat is about as powerful as the unharmonized data with a scanner covariate. So it's preserving power while also controlling type 1 error better. And these are just sort of the estimates across all regions of interest. And you can just sort of refer to the paper for more details on this. But now I'm going to just, and here's the final estimates across the brain. And we did find, more atrophy occurring in medial temporal regions as expected. But yeah, now I'm going to just briefly go over some other extensions of combat that I mentioned. So combat GAM in the generalized additive model framework. So it's often convenient to have, a more flexible model for modeling age trends in like, developmental trends in the brain. And so this lifespan data set is very large, over 10,000 scans. And each participant contributes, only one scan here. And these are pooled across, many studies. And they were able to, harmonize the data using this combat GAM. And there's a very cool, shiny app you can check out at this link where you can actually upload a data set and harmonize it to this lifespan data and compare against, this data set. And for combat, I don't know how to, okay. For the combat, generalized additive mixed model, we're just going to incorporate, we have a smooth function here, and we can incorporate random effects. So when we have multiple measures, within a subject, and here's an example of data. I was comparing here the GAM with the GAMM4 functions. And this is the same ADNI data set, but there really wasn't much of a quadratic effect in this data. So for this, for developing this combat GAM, I just added a quadratic effect to illustrate but yeah, you can see again that a lot of the variability is removed, the scanner-related variability after harmonization. COVBAT corrects covariance batch effects. So it has several steps. So the first is just to do standard combat on the data. And then step 2 harmonizes the covariance of the combat adjusted data. And it's harmonizing the variance of the principal component scores. And you can see that on the top row here, we have unharmonized across 3 different scanners. And the bottom row is the COVE-BAT adjusted after, the harmonization. You can see, the data should look more similar. And this is papers up on bioRxiv right now. And Andrew Chen performed these experiments where, trying to predict the scanner from the unharmonized data, and then from COMBAT adjusted data, and then the COVE-BAT adjusted data. And it becomes harder to predict the scanner after applying COMBAT, and then even harder after applying COVE-BAT, which is good. So the scanner effects are being removed there. And also we're seeing a better ability to detect AD versus controls, and better ability to distinguish male and female sex. And this is in Andrew's poster number 31, where this is specifically for harmonization of functional MRI data. And, they showed that there are scanner effects in the detection of the brain community structure that become, these can be removed to some extent using this block COMBAT, or the functional connectivity COMBAT, and then we have functional connectivity COVE-BAT. So yeah, I would suggest checking that poster if you're interested in learning more about that. Because I think there's been less work really studying functional MRI data, harmonization of functional MRI data. So just to sum up, the advantages of COMBAT, and I didn't discuss really a lot of other harmonization methods, but there are, let's say, deep learning based methods or other methods, but COMBAT is, one advantage is that it's very interpretable and fairly simple to understand. And it's also flexible. I mean, you can adapt it to any type of model, really. It seems it doesn't require an overlap cohort. So sometimes some methods require you to have a certain set of participants scanned on both scanners, or more, every scanner. But COMBAT can deal with the fact if you don't have overlap, but hopefully your covariates are balanced. That's important. Also, the robustness to outliers in small sample sizes. Maybe you could consider disadvantages. You need to carefully specify the model and account for confounders, and preserve the biological variability of interest. So sometimes it's a concern if you're like doing clustering, or you have unknown biological variability you want to preserve in the data. You might think this could be problematic, but there are ways to train COMBAT only using a control sample just to estimate the scanner effects. And then you can apply the model to, let's say, a psychiatric, sample where you may not know, what biological variability you want to preserve. So you can just leave them out of the harmonization. Another thing is that the harmonization should ideally match the final model exactly. So you might have to harmonize multiple times if you're doing multiple models. And here's some references I'm happy to share later if anyone's interested. And yeah, thanks very much to the organizers, to Dana, to my postdoc mentors, Kristen and Taki, and the Pensive group, Andrew and Hao Cheng for some slide content. Nick and Phil helped with the ADNI data set. And yeah, I think that is it. So I'll just got